Okay. So, lots of different ways to get to the lysosome. There's two ways that are constructive, and other different pathways are destructive. So you have to have a couple of ways to get cargoes into the lysosome to run its business. Okay. If you are going to make a lysosomal protein that's a membrane protein, you use this pathway. You bud off from the Golgi using clathrin-coated vesicles. You use AP1 as your assembly particle, as opposed to AP2 coming from the plasma membrane. So you bud off, you go straight to the late endosome, and basically here's a good rule. If you go to the late endosome, you're going to the lysosome. They're really different maturation states in the same compartment. So all the membrane proteins go this way, and then if you are a soluble enzyme that functions in the lysosome, and there's dozens and dozens and dozens, then you have to go this way. You have to go to the early endosome, back to the late endosome, and then to the lysosome. There's a very good reason for doing it that way. Okay, so let's look at some examples. Here's a really good example of a membrane protein that must go to the lysosome. The lysosome functions at low pH. So to get that low pH, you have to have a VOB1 ATPase. We know how that works. That's actually going to be on quiz 2. And so the ATP domain is the V1, and it faces the cytoplasm, which it must to be able to pick up ATP. And so it's going to split ATP, and it's going to drive protons uphill into the lumen of the lysosome. Okay? Now, these are just dozens of categories of proteins that are soluble, and they get there by a slightly different route. And so what happens is when the stuff gets into the late endosome, it will eventually mature into the endolysosome, or it will actually fuse with the lysosome. This kind of always reminds me of a sort of a Japanese Hello Kitty kind of thing. And then eventually becomes North Korea. So if you go here, you will be destroyed, and usually by shooting missiles, they fall back on yourself. Okay, so that's that pathway going that way. Let's talk about the ones that go to the late endosome as a destructive pathway. So those are constructive pathways to build the lysosome. This is things that you're taking to the lysosome that must be destroyed. And there's 3 different pathways. The one that you're probably most familiar with is the one we alluded to. That is, if you endocytose things from the cell surface, like LDL particles, to get the goodies that are in an LDL particle, which is fatty acids and cholesterol, you want to send it to the lysosome to digest the protein and then release the cholesterol, and the cholesterol will diffuse into membranes. So the way that works is you go from inside the cell to the early endosome, to the late endosome, and then if you reach this, you're destined to go to the lysosome. Okay. So those are cargoes coming in from the cell surface that need to be metabolized or destroyed. Okay. The second one is phagocytosis. So if you're a large cargo, like a bacteria, that said maybe this is a macrophage and it's attacking and cleaning up all the bacteria, then it's going to use a fundamentally different process called phagocytosis. And I will show you the difference between this type of clathrin-mediated endocytosis and the mechanism of phagocytosis is quite different. You're dealing with a huge package. So it wraps its arms around that, pulls it in, it becomes a phagosome. It's a single bound membrane. Okay, it's not double membrane bound. But that's because this cargo that's the thing that it's picking up, is a bacterium. So it has one cell membrane contributed by the host cell. And so the phagosome then fuses with the late endosome, and then of course it becomes the lysosome, because that's the dead. So the third thing is a really interesting process that's discovered many years ago, but now it's starting to become very important, because it turns out to have roles in cancer and all kinds of different processes. It's called autophagy, or if you want to be really snotty about it, autophagy. Okay, and what it means is self-eating. Okay, so what happens is our mitochondria, our ER, various organelles get old. Okay, how would a mitochondria become old? What would cause it to become old, where it's no longer functioning as it should be? Is there anything really dangerous going on in the mitochondria? Yeah.
From the electron transport chain?
Yeah, those are high energy electrons, and oftentimes when they get the complex 4, which I'm sure you have memorized, it hits there, it's supposed to go and oxidize or reduce that oxygen to the water molecule. Sometimes those electrons they leak out of complex 4, and they form free radicals, they form hydrogen peroxide. Those are very damaging. Now the problem with mitochondria is the DNA is very exposed. It's bacterial DNA, so there's no nucleosomes to protect it. So those free radicals come in and modify the DNA. They actually oxidize the lipids in the mitochondrial inner membrane, and so you're going to poke holes. What happens if you poke holes in the mitochondrial inner membrane? What's that going to mess with? Yeah, the proton gradient that you established because you sent those electrons through the electron transport chain. You pumped out protons, and you want the protons to go back through which protein would you like it to go through? You want to make it. What are you going to do with that proton gradient? You can't do anything directly with it.
Go make ATP.
Yeah, you're going to make. So you send it back through the ATPase. You're running an ATPase backwards by running it down, the protons down its electrochemical gradient, and so you're going to spin it around as a turbine engine, and it will make ATP. Okay, but if that proton gradient is leaky, right, you're going to lose some of your energy, and you won't make as much ATP. So old mitochondria are bad news. In fact, there's actually hypotheses for type 2 diabetes that had to do with aging mitochondria. Okay. Anyway, so if you have an old mitochondria, the way to get rid of it is you have something that really looks like part of the ER. So you have a double membrane that forms, so it's like a sac of the ER wraps around the smooth ER, wraps around the target, damaged mitochondria, and forms an autophagosome. So now this has got two different cell membranes to go through. There's two different membranes there because it's like wrapping the ER all the way around the nucleus. So you can think of this autophagosome as being like the nucleus, and the ER is the nuclear envelope. So it's two different lipid bilayers to get in here. So this whole thing then fuses with the late endosome. Where's it going to go? It must go to the lysosome. So all of these destructive pathways must fuse with the late endosome and then destroy it. So what's the relationship to cancer? Well, what happens is a lot of times the cancer cells try to feed themselves, and they build a network of blood vessels. But the blood vessels are really crummy, and oftentimes they don't provide enough nutrition to the cancer cell. So to survive, the cancer cell will do anything, including eat its own parts. And so it stays alive and resists destruction by autophagy. Okay? So it's a way of the cancer cell staying alive. When people undergo fasting, sometimes what happens is the repair mechanisms take over. If you don't have a lot of nutrients come in, the first way the body responds is to actually start repairing things. It's one thing you can do. If you're not getting enough energy to build, you can at least repair. And part of the things that go on during fasting that's actually very good for you is autophagy. So you get rid of all the damaged stuff. And then when you go back to having enough nutrition, then you can move on to constructing new things. Okay? So autophagy is a really interesting area right now. Okay, so all right, so let's take a look at this. This is just a problem that's related to membrane or vesicle trafficking. And this is not a clever question. What I want you to do is just read this and find out, critique it. Tell me what's wrong with it, because it's the kind of question that can be asked on an exam. Not quiz 2, but quiz 3. Okay, if you happen to be near your group members, then take a minute and just talk about what you think. There may be several things wrong with this. So.
So that helps
Let's just say what's correct about this. What part of this is correct? Or is any of it? Group 10? Since some people volunteer. Just take a shot. Where's group 10? Group 10. Okay. That's hiding. Oh, there we go. Okay, good. 10, 10. Okay, so what's good about this? Or is it all good? Is the GTPAs involved in the directionality of where that cargo is going to go? Is that part of the story? Well, let me ask a different question. If you want to go to a particular destination, do you need to use a particular type of coding? Yeah, okay, so that part's correct. And to have the right coding, you got to have the right GTPA. So that's absolutely correct, okay? So if it buds off, okay, and if it buds off and it's going to recruit these units, and this is all correct, okay? The problem is, the first thing you have to do, that's our rule, if it buds off, the first thing that has to happen is it must uncode. Is that what this says?
No.
No, it says that the gap, which is going to turn on the uncoding process, is actually located in the destination cistern. Okay. So that's clearly wrong, right? So the gap is actually part of what? Where is the gap that's going to cause the encoding? Pardon me? Yeah, it's part of the COP2 code. Yeah, it's actually, so it's SEC 1331 activates the gap activity of that SEC 2324. SEC 2324 is a gap. It's a GTP activated protein that kicks SAR in the butt and causes it to split GTP. If it splits GTP, why is it it's gonna dissociate from the vesicle. Why? What is the GTP state controlling as far as SAR is concerned? Pardon me? The landing gear. So the landing gear is going to retract, so it has no way of being a lipid anchored protein anymore, and so it's going to retract, and it's going to fall apart, and its affinity for all the other stuff that's holding on to it is going to just fall apart, so it will uncode. Right? But that happens because of the coding, and actually the curvature that it experiences. So it has nothing to do with the target rendering. So that's what's wrong with this. Okay, so let's keep this, trafficking is a big deal because it's got so many components. So speaking of that, we want to talk about this issue right here. pH is a big deal in how it changes through the secretory pathway. Not only just to make the lysosome really acidic, but there's a change in pH as you approach the lysosome from any direction in the cell. And what I mean by that is you start out in the ER, that's where the proton pumps, the VO-V1s, are assembled, and so they start to pump, right? And so that starts to be acidified from pH 7, it starts to drop. Every Golgi stack that you go through starts to drop in pH. So by the time you reach the trans-Golgi network, the pH is now no longer 7, it may be 6.8, 6.5, okay? And then if you butt off into the early endosome, it's even lower, it may be as low as pH 6. Then if you go to the late endosome, what's your guess about that pH? Even lower, it's like pH 5. And by the time you get into the lysosome, it's 4.5 or lower. Okay. So, and if you're coming from the cell surface, the cargoes that the endosomes that come off the cell surface form, those are pretty much, pH of 7 because it's extracellular fluid. But then it starts to drop because you actually have the proton pumps that are included in the endosome, the early endosome in particular, so the acidity starts to drop again as you go towards the lysosome. All right, so it's a big deal. So let's talk about how do you get something all the way from the endoplasmic reticulum that's a soluble lysosomal enzyme. What is the targeting mechanism? We know about the coding process. We've already talked about that. It's COP2 that leaves here, gets it to the Golgi, and then in the Golgi, you don't need a lot of trafficking, right? The cargo is going to move because the cisterna are moving, the cisternae are moving along railroad tracks that are the cytoskeleton, microtubules, and they're little motors that are carrying the cisterna forward anterograde. Okay. So we're going to get to the trans-Golgi network, and then it has to have a zip code. So what is the zip code that's going to get it to go here? Go to the endosome, go to the late endosome, and lysosome. Okay, so that's the issue. Well, this is the zip code. It is a mannose 6-phosphate. Now, if you're going to guarantee that a sugar residue on a glycosylated protein, so this is the lysosomal soluble enzyme. If you're going to guarantee that it gets a mannose, which type of glycosylation must you use? N or O? And why? It has nothing to do with the nitrogen or oxygen that's attached to it. It has to do with the type of thing that's attached always to one of those types. Which one is guaranteed to have a mannose? Okay, let's think about the two. The differences in the two. The O-linked, how is it different from N-linked? You attach sugars to oxygen, right? But how do you do it? You do it one at a time. You put a sugar, you put this, and it doesn't need to be a mannose. It can be glucose, fructose, it can be all kinds of weird stuff, right? But if you're going to attach an N-glycosylated sugar residue to a protein, you have to attach this tree, oligosaccharide tree, and it has high mannose number in that oligosaccharide tree. So it must have a mannose. So it's not necessary that you have to do all glycosylation using N-glycosylation. You can have both. You can have N and O glycosylation of the lysosomal protein. But you must at least have some N-glycosylation so you're guaranteed to have a mannose tree. Okay. And that's where you're going to stick a phosphate group at the 6th position. And this is the zip code to get you to the lysosome. So now we need to explain how do you get the zip code, where is it added to the envelope, and then how does that guarantee you go to the lysosome? So those are different questions. So the first one is how do you get the zip code on there? It's not attached. It is not attached in the ER. It's attached. There's actually two places where this zip code is formed. The first is the phosphorylate this glycoprotein on the sugar residue at the cis-Golgi network. That's where an enzyme lives that is N-acetylglucosamine phosphotransferase. That's a really ugly word. This enzyme lives in the cis-Golgi network. And what it does is it has a binding site that recognizes that, that's a lysosomal enzyme, and it has a binding site that binds to UDP, N-acetylglucosamine, and then it's going to transfer a phosphate, but it's got a little piece left on it. It's got N-acetylglucose attached to that phosphate. So the zip code is attached in a hidden message. It's there, but it's masked by that N-acetylglucose sugar residue. That keeps it masked, okay, as it moves through the Golgi. And when you get to the place where it's got to make a decision to leave in the trans-Golgi cisterna, that's where an enzyme is that chops off that little N-acetylglucose right there. That's chopped off. That then reveals the mannose-6-phosphate tag, and the trans-Golgi network goes, oh, okay, I'm going to send you to the early endosome. And it does that because the trans-Golgi network has a mannose-6-phosphate receptor. So the enzymes to put that tag on are in the cis-Golgi network. The enzymes that untag it are here in the trans cisterna, and then sorting occurs in the trans-Golgi network. Okay? So you unmask it. There must be, in this membrane, a mannose-6-phosphate receptor. And we've been looking at it a lot, actually. So this is where it must be. The tag is put here, but it's covered. It's uncovered here in the trans-Golgi. Okay. Then when you get to the trans-Golgi network, the zip code is expressed, and then you need a cargo receptor, which is the mannose-6-phosphate receptor, that recognizes the cargo, and then it recruits clathrin coating. Now, how does it do that? You've got a cargo receptor, mannose-6-phosphate receptor. The characteristics, it must have at least a single pass membrane protein so that there's a cytoplasmic domain that can be recognized by the coding machinery. Right? What is it that's actually going to bind to that cargo receptor cytoplasmic domain? What's going to bind to it? This thing is actually, this is what's going on in this membrane. Okay, we use this to sort of generically talk about the parts, Mickey Mouse and all that stuff. So ARF must come down, it must lower its landing gear, it must recruit Mickey Mouse. In this case, it's AP1. Okay, and then the alpha adaptin is going to bind to ARF. The alpha adaptin is going to bind to phosphatid PIP2, and what's the third thing the alpha adaptin is going to bind to? Well, what actually binds to the cargo receptor is the mu1 component, right? The mu1 component. So that's actually the part of Mickey Mouse here that recognizes this thing. Okay, and then the beta adaptin is going to recruit what? The heavy chain of clathrin. And so all the parts come together. So that's how this, you got to have a cargo that matches it's recognized by the cargo receptor, which is the mannose-6-phosphate receptor, NPR. So when this thing gets loaded, the clathrin coating forms, this thing buds off. And the first thing that's going to happen is it's going to uncoat, and that will, so it uncoats immediately. And anytime it's in a vesicular form without being in an organelle, so that's an organelle, this is an organelle, that's a transport vesicle. That's the generic term for anything that's just a vesicle that's going somewhere that has not got to its organelle yet. So it's in a transport vesicle that's destined to go to the early endosome. Now, what makes it go to the early endosome? Several different things. And this is going to congeal our knowledge from what we talked about last time, vesicle trafficking. What must this vesicle be displaying? What types of protein? Does it just need the mannose-6-phosphate receptor? One protein? Yeah.
Well, it's not a protein, but it'll have the phosphatidylinositol that's phosphorylated at that place wherever it is. Right?
So this must have modified its plasma, its lipids to be the appropriate one that's going to fuse with the early endosome, right? What phosphorylates those lipids? Phosphatidylinositol something kinase, right? You don't know if it's 3 or 5. You know, we can go look it up, right? So the lipids have to be correctly phosphorylated. What causes that kinase to sit down and phosphorylate the lipids? Okay, you can look that up. Okay, so we get here by some magical process that you're going to look at, and it fuses with the endosome. And then what happens is it's stuck very tightly to the cargo receptor. So the lysosomal enzyme is bound very tightly to the the mannose-6-phosphate receptor. How do you get them to uncouple? Okay, and why should you uncouple them? So what it's going to encounter is a fairly acidic pH. That's what releases the cargo, which is the enzyme, from the receptor. We want the cargo to go to the lysosome. We don't want the receptor to go because we want to reuse it. So what happens is when it hits this low pH, the cargo dissociates. The lysosomal hydrolase is going to go from the early endosome to the late endosome. And then you know the rest of the story. If it goes to the late endosome, it must go to the lysosome. But what we want to do is we want to re-grab that receptor before it leaves the early endosome and goes to the late endosome, and it actually is retrieved back to the transgolgi network. And this is a special coding mechanism that we're not going to discuss, but it's got its own properties. So it recruits this back to the transgolgi network, and we can do this over and over without destroying and wasting energy. Whereas the cargo that we want goes to the lysosome, and this builds the lysosomal function because that's one of its enzymes that chews up stuff, right? Okay. And before it can do that, this phosphate is actually removed as well. Okay, so that's one example of how changes in pH are going to tell you when to release stuff and when to keep things together, right? Any questions about that other than all the stuff that you didn't know? I'm sure you'll look at it, okay? Probably not before the test. All right. So, we've done this trip here. Early endosome, late endosome, lysosome. Now what we want to do, let's just, I'll mention one particular medical condition that's involved in this or related to this. Some people, let's see, they have a genetic deficiency for this enzyme. They can't make this enzyme. And so they have what's called eye cell disease. It means inclusion, cells that have inclusions. Okay? And so why are those people going to have inclusions? Just blobs of stuff that should have been destroyed, they're just going to be sitting there. And it's actually a fairly discrete population of fibroblasts that do this. Not all of their cells do. So that means that there's some differences in trafficking to the lysosome. But these people have problems. And you can see if you can't put this tag on the lysosomal enzyme, what is going to happen to it? Is it just going to stay here? Where's the default place that it's going to go? If you have no tags that divert you, what's the default position? Outside here, it can be secreted outside the cell. Okay? But the problem is those enzymes never make it to the lysosome, so there's stuff that's coming to the lysosome that can't be digested. So the inclusion bodies are the lysosome, an ineffective lysosome. It builds up and it kills the cell. Okay? But in the liver, this lysosomal trafficking works just fine. So that implies it must use a slightly different zip code. Okay? So eye cell disease is a lysosomal problem. Okay, so let's talk about endocytosis as the book describes it, and not everybody calls this endocytosis. Okay. Different textbooks do different things. So your textbook thinks that endocytosis is all the stuff we just talked about. Clathrin-mediated endocytosis, phagocytosis, all of that is a type of endocytosis. And so it says there's two types of endocytosis. There's phagocytosis, which is the eating of big things. That's clearly very different from pinocytosis, which technically means cell drinking, but there are two types of drinking. One is the one that we just talked about, clathrin-mediated endocytosis from the plasma membrane. That's also called receptor-mediated endocytosis. And then there's clathrin-independent endocytosis, which involves constitutive bringing in endocytosing the plasma membrane with a little fluid in it. Why do you need, if you're a secretory cell in, say, the not the lysosome, but the pancreas, it secretes lots of different enzymes. So exocytosis is going on all the time. Why do you need this to be going on all the time, too? To keep the cell
from shrinking, so it maintains its size.
Well, actually, it's in danger of getting too big, isn't it? Because you have the right idea. It's actually the correct idea is that the homeostasis of cell size is correct, but it's different for different things. If you have a cell that is producing lots of secreting stuff, every time a vesicle fuses, you release carbon, right? But you also increase the cell membrane by that amount of lipid. So the cell's going to grow and grow and grow, and so a pancreas cell would get huge if it was not for that process. And it's just constitutively to bring stuff back in. Okay, so there's a net homeostasis of membrane size and delivery. Okay, so we've talked about all the details here, so we don't need to go into all this. You know, clathrin and adapter proteins, we've already done that trip. Okay, let's talk about a couple of specific things in regard to LDL and a couple of examples of endocytotic clathrin-coated endocytosis. So we're going to talk about this in particular, but not all the details of how you get it to come in. We've already covered that. Okay, so let's go back to this. And so we have lots of different kinds of coatings, and we haven't actually exhausted all of the coating types. So this is a clathrin coating that we're familiar with. It's budding off at high resolution, and so but there are other types of coatings that are covered in cell too, because we don't have time to do it. These are called caveoli. They're like little cavities that form. And different types of cargo from the ones that we're considering actually use a different coating type, and they look sort of like cabbages. That's what their coating type. Instead of, see, that's a clathrin-coated vesicle that's coming off the cell membrane. These are caveoli. They look like cabbages. So there's a whole different set of coating mechanisms for different types of cargo. So we'll leave it at that. The one we're familiar with, though, a classic cargo that we use, receptor-mediated endocytosis, clathrin-coated endocytosis, is the LDL particle. And in case you're unfamiliar with what an LDL particle, low-density lipoprotein particle is, it's actually sort of a vehicle for carrying lots of cholesterol. It's got fatty acids. It's not a bilayer, it's a micelle. So it's only got one single layer of lipids on the outside. And then it traps a lot of cholesterol. And in this thing, you must have this big lipoprotein protein. It's called B100. And this protein is necessary to be recognized by the clathrin-coated endocytotic machine. So the receptor that's going to recognize this looks for this protein, not for the lipids. Okay. And so we want to take this in because we use the cholesterol to what? Make membranes. What if you're a cell in the ovary? What are you going to use the cholesterol for? What comes out of the ovary every month? not the egg, the hormones, right? Your steroids. So it's progesterone, it's estrogen, it's a testicular cell, it's going to be testosterone is made from cholesterol, and this is the raw material to make steroids. And the adrenal cortex that makes glucocorticoids needs to just eat up all this LDL. And that keeps your blood levels of LDL low. What happens if your LDL levels are too high? You get a heart attack. You do. All right, so you need this system to constantly pull that stuff in, and the way you do it is to use this mechanism that we've already talked about, okay? So we're not going to go through all this, we've already talked about it. But the key thing is, it's really critical. This is the cargo receptor, the LDL receptor. The rule is, if it's a cargo receptor, it must have at least one transmembrane alpha helix. And it also must have a consensus sequence in the part that's facing the cytoplasm that is recognized by AP2. AP2 is the Mickey Mouse that works with the cell plasma membrane, okay? And we know what that consensus sequence is. It's asparagine, proline, something else, and then tyrosine, okay? And so that the Mickey recognizes that the mu subunit actually binds to that based on that consensus sequence and pulls this thing in, right? Because it initiates the Clapton coding process. So it comes in. Now, here you have a problem. So you uncoat. What is this called? What kind of vesicle? It's not coated. It is generic. It's not this is an organelle. That's an organelle. That's an organelle. This is Ransford vesicle. Okay, so it's gonna fuse. It's displayed its targeting information. It's going to use all that magic that you're going to talk about and look up. Okay, and it's going to fuse with this thing. Now, the problem is, if you don't dissociate the cargo from the receptor, what's going to happen when the cargo goes to the lysosome? You're going to chew up this guy. You're going to waste a receptor. And this is something that needs to happen constitutively to keep this cell happy, all right? So if you're going to use the thing over and over like you did with the mannose-6-phosphate receptor, you better recycle it. So recycling works differently here. You come to the endosome, but it's not the same principle. What kind of pHs are you going to encounter in the early endosome? Acidic, right? Okay. And so that causes this to uncouple. It releases the cargo, and this is actually trafficked back through a transport vesicle to something called the recycling endosome. And if you go to the recycling endosome, you're almost sure to spin off a vesicle that goes back to the cell surface. That's why it's called that. Okay. You go to so just like if you go to the early endosome to the late endosome, you're destined for lysosome. If you go to the recycling endosome, you're destined for the cell surface. So that's why this is a special compartment that's different from here. This is a decision tree right here. You make a decision. Now, we actually know the mechanism by which the acidity causes that to uncouple, and it's kind of interesting. Maybe not to you. All right, so what happens is, let's draw a really large vesicle. Is this in focus? Somewhat, okay. Large vesicle, okay. And so let's draw the machinery. This is the cargo receptor. So this is the LDL receptor right there. This is cytoplasmic, okay? And so it's got that sorting signal, which is NP, X, Y. And the X means it can be any amino acid except for proline. And the Y does not mean Y, it's another variable, it means it's tyrosine. So you have asparagine, proline, X, and then tyrosine, which is a bulky, hydrophobic amino acid. So that's what the coding machine is looking for. That's what at the cell surface. So AP2 recognizes that. But what's going on with the other side that's actually holding on to the cargo? First of all, you got this really neat little piece. It's a motif. It's sort of a cup-like. And I'm trying to draw sort of a concave surface here. This is called a beta propeller. Okay? You can imagine a beta sheet that's sort of arranged in a fan shape, like a propeller and a fan. That's the way this is arranged. And so that's the motif that actually holds on to a little piece, okay, that's sort of dangling like this, and it's got negative charges. This is the ligand binding domain. Of the LDL receptor. It's got negative charges, okay? And it holds on to the LDL cargo this way, like that. Now what happens is when the pH drops like this becomes more acidic, you've got more proteins, and the beta propeller picks up positive charges. If this is the early endosome, you got low pH, 6.5 or lower, that those protons protonate the surface of the beta propeller, and guess what happens? This thing would rather stick to the propeller, so it does. And then that causes the release of the LDL part. So that's how the change in pH physically causes that protein to release the cargo. Now, once you've done that, the cargo receptor can be shipped to the recycling endosome, and then this cargo is going to the late endosome. So they split ways there. And so you retrieve the cargo receptor to be used another day. Okay. Any questions about how that works? So we know a lot now about how pH is regulating the way cargo is disposed of. Let's take a break here and we'll talk about some other. Okay, why don't we get started again? Just another medical moment related to LDL particles. There are people that actually do have mutations in that consensus sequence of the LDL receptor, and they have a condition called familial hypercholesterolemia. That means high levels of cholesterol in the blood, and they go way up here. Okay? I'm not sure. They must have to take really high doses of statins, but if they're undiagnosed, they develop heart attack, atherosclerosis, and have heart attacks by age 20. So it's really important to screen for this and get them on cholesterol-lowering drugs. But the mutation is in that consensus sequence, so that Mickey Mouse AP2 does not recognize the cargo receptor and pull it in. So that's what happens. We're going to kind of skip phagocytosis and come back to it, but one of our learning objectives is to distinguish endocytosis mechanisms from phagocytosis, and it's quite easy to do. So we'll move to this part here, and that says, well, what happens to things that are endocytosed? Well, you know that they can, if you don't do anything else, they're going to go to the lysosome. If you don't retrieve them and recycle them, then the goal is to go straight to the lysosome. So that's the, what's the term? That's where it's going to go if you don't do anything else. Okay. Specific proteins, there's a decision made about whether to keep it or not. And one of the decisions is based on what the function of that cargo receptor is. And we've talked about some of these. So the LDL receptor, you know that's a constitutive process. We want to use it over and over. So we're going to recycle this cargo receptor. And then we have a class of what I call dangerous receptors. They're very useful. Epidermal growth factor is a very important receptor that when you activate it, it causes cell division. And so this stuff is secreted in your saliva, but if it's overexpressed, you can get salivary cancer and GI cancers. And then of course, epidermal growth factor are overexpressed in breast cancer a lot. Okay, so this is a dangerous receptor to leave active. And so what happens typically is it's not recycled. So you activate the signal. So EGF binds to the receptor, and it's really not a cargo. It's a receptor. The primary goal, if the goal is signaling, it's a dangerous receptor. And it may be recycled for 1 or 2 rounds, but typically it's going to be pulled off the cell surface and silenced, okay? And to do that, all you have to do is send it to the lysosome, okay? So dangerous receptors. And then transferrin receptor, what's that doing? That's pulling off transferrin, getting iron to come in, and that's regulated at the level of the messenger RNA, as you know. That's going to be on this test, quiz 2, on Tuesday. So this is one of those receptors that you probably would recycle, right? Because you want to keep iron coming into the cell until you quit making the transferrin receptor by those mechanisms that we talked about. Okay, so let's go and look at some of the differences in these fates. Done this one. So our goal is to understand the different fates of these cargo receptors, and then we'll distinguish between phagocytosis and endocytosis. All right, this is a really interesting experiment, and it shows how the decision can be made inside the cell with dangerous receptors and with one that's a constitutive receptor, like the transferrin receptor. So here's the labeling. So we can label different proteins with different colors so that we can follow them. So in this example, one of the cargoes is the opiate receptor. That's obviously what binds to endorphins, and if you happen to snort cocaine, that's what binds to that. So that's a signaling receptor, and it's dangerous, isn't it? So it's like the EGF receptor. It's not a cargo receptor. Its job is not to bring more opium into your cell. It's to turn on if it detects the cargo. So that's a very different thing. So we want to destroy that, and that cargo receptor is labeled in green. It's labeled with a green fluorochrome. Okay. Now we have transferrin in this figure. The transferrin is labeled with red. Okay. And if you remember your spectrum from physics, you have Roy G. Biv, red, orange, yellow, green. So what's halfway in between red and green? It's halfway in between orange and yellow. Okay. So if both of those proteins are in the same compartment, it will turn yellow. If they're in independent compartments, it will be green or red. Okay. So what happens is we can put the ligands in here. We've got the labeled receptors, and we let things run for a while. And what you notice is what actually turns green is the late endosome. That's where all the opiate receptors go. Now, to get to the late, so we want to destroy the opiate thing so we don't become too wild and crazy, right? And so you destroy it. So if it goes to the late endosome, where's it going? Lysosome with its cargo. Okay, you don't uncouple them or anything. Now, so it goes there. That's why the late endosome is green. But to get to the late endosome, what does it have to go through first? It's coming from the cell surface. So all those cargoes and receptors have to go someplace first. They come in by clathrin coating, right? AP2 comes in. They have coat, becomes a transport vesicle. It's going where next? That place where the pH is low, kind of low. It's not as low as the late endosome. Pardon me? Yeah, early endosomes. That's where everything, so that's the fun. You've got to know that. When it comes in from the cell surface, it's got to go there. So that compartment, in some cases it might be, you might catch one that's red or green, but there are a lot of them are going to be yellow. So that the yellow compartments here are early endosomes. Okay? The late endosomes can be nothing but green, because that's where the opiate, the dangerous ones go. And the recycling endosomes are exclusively red, because that's where transferrin is going. So transferrin comes in, clathrin-coated vesicle, uncoats, becomes a transport vesicle, goes to the early endosome, makes a decision, releases its cargo. What's its cargo? The transferrin receptor is releasing what? This is on test 2, people. You were going to have test 2 today. So you should know that. In the word itself, transferrin receptor carries transferrin, right? Bound to, you're not after the transferrin, you're after iron, right? Bound to, so that's a tricky situation itself. But what happens, so the transferrin comes in and goes to the early endosome, the transferrin receptor then goes to the recycling endosome. And the rule is if you go to the recycling endosome, you go where? Back to the plasma membrane. So there's these rules involved. And so anything that's red is exclusively red is a recycling endosome. So you can tell by the different colors. So it's kind of a neat experiment. Okay, so we have a problem though with this pH and getting rid of transferrin. Is a little bit more complicated than it first looks. Let's get this. Okay, so we're going to change. So we're going to take. Let's focus on transferrin since it is on the test on Tuesday. Okay, so we want iron. This is about dead. Okay, and so iron is toxic floating around, so it binds to transferrin. And that happens in physiological pH in your intestine and your bloodstream. And then this eventually goes to the cell that needs the iron. And so you have what? A transferrin receptor. Okay? And so the rule is if iron is bound to transferrin at physiological pH, it has a high affinity for that receptor. So what will happen is that receptor will bind to the transferrin, which is this blue blob, and the iron that's attached to it. Okay. Because it's at pH 7.0. So it goes into the cell, it goes to the early endosome. This is kind of bubbly looking thing, so this is the big EE, not electrical engineering. And so what happens is this gets there, and you know that the pH changes. Okay. And so what's going to happen? Is the transferrin going to dissociate with the iron, or is something else going to happen? You want to retrieve, so here's the rule is that you want to retrieve not only the cargo receptor, this thing, but you want to retrieve the transferrin and keep it going and recycling. So all we want to do is release the iron. So at low pH, 6.5 or 6.0, the iron is the only thing that dissociates. So the rule is at low pH, iron dissociates from transferrin, but the transferrin does not dissociate from the cargo receptor. And that's really cool because the iron can then be sent to the lysosome, and then the iron is transported to where it needs to go, like to the mitochondria. Okay? So it's okay for the iron to go to the lysosome, but it's not okay for the cargo receptor or transferrin. So it's actually shipped to, strangely enough, the RE, the recycling endosome, right? And what's the pH going to do as it approaches the plasma membrane? The pH is going to increase. Okay. So we have the cargo receptor, we've got transferrin, and what happens is when this sends a transport vesicle, it fuses with the plasma membrane, and what this looks like is here's the cargo receptor, like that. And the rule is if transferrin has no iron associated with it, at physiological pH, transferrin will dissociate. That's a different rule. So at physiological pH, as long as iron is not there, the receptor will let go of the carrying protein, which is transferrin. And it goes out hunting for another aberrant iron atom, or ion in this case. So the relationships are different between cargo and receptor depending on what the cargo is. And I did this on purpose because it is one of the more complicated systems. If you labeled, you did this experiment and you labeled the LDL receptor and the transferrin receptor, would you see anything of any color in the late endosome? No. But you'd see the early endosome would be yellow, and the recycling endosome would also be yellow because they both go to the same place. Okay, can somebody have a question? Or did I? Or you just go on? Okay. All right, questions about that? Now, the emphasis on test obviously will not be this kind of trafficking, but the questions about how you upregulate or downregulate transferrin receptors and blah, blah, blah will be fairly. Okay. All right, now, there's something, so there's still some more cool things that you can do. So the fate of something endocytosed, you can take it and you can destroy it with a lysosome. You shouldn't say destroy, you should say metabolize. You can metabolize iron, you can metabolize LDL. You can take it and you can recycle it, and then you can do this. And this is called transcytosis. And my little pointer is still over here. Transcytosis. When would you like to do this? Okay, so this, you're going to want to do this type of deal in nursing mothers. Okay. So what happens in a newborn is nursing, and what they get for the first few months or a certain amount of time, they can't make their own antibodies at an appreciable rate. So what they do is they take the mother's antibodies provided for them in mother's milk, and the GI system doesn't destroy it. It's kind of weak. And what it does is it actually takes those mother's antibodies and retrieves them, sends them into the blood, and gives passive immunity for that newborn. Okay? So how does this complicated thing happen? This is the mother's immunoglobulin. It's shaped like a Y, right? You turn this up. This is IgG. Okay, immunoglobulin looks like a Y. And you know that there are omega loops here and here that detect antigens. And so this little part right here is called the FAB fragment, the antigen binding portion. So the F stands for fraction, the AB stands for antigen binding. And so this is the FAB fragment. What's this part? Well, it's called the FC. That C stands for crystalline, because when people isolated these things, they put them in the refrigerator to crystallize. Okay? So it's called the FC fraction. But you can also think of that as being constant. This is not the variable portion of this, it's the constant region. So that's what I like, that C really stands for that. So this little portion is the cargo that's going to be recognized by that cargo receptor. So in the newborn has an FC receptor on its apical surface of the intestine. So that binds to this. AP2, Mickey Mouse sees it, brings it into the cell, platinum coating, and you know that story. Okay. And so where does it go? It has to go to the early endosome. And what happens is it encounters this low pH, but this particular cargo and receptor don't let go of one another. They stay together. Okay, now normally that would be a bad thing, because this would then send a vesicle, and this transport vesicle would go to the late endosome and would destroy it. That's not the point here. We want to put this cargo in the bloodstream. So it's got to cross the epithelial cell, because remember, this is the Bart Simpson side, and there's a tight junction here. So the antibody can't leak into the bloodstream. We don't want that to happen. Okay. So what happens is the transport vesicle is targeted to the recycling endosome. And the rule is, remember, if it goes to the recycling endosome, it's going to be sent back to the cell surface. But this recycling endosome is smart, because it says, well, I have two plasma membranes. I have to be able to pick one. And it knows, because of the RABs, which is that thing you forgot earlier, the RABs is going to tell it to send it to the basolateral membrane. And when this thing encounters the neutral pH, this is slightly acidic, this loses affinity for the receptor at physiological pH. So again, pH is determining whether you go or whether you hold hands or what you do. This is acidic because it's in the intestine and acid is coming down the pipe. This is not acidic, that's pH of 7. So it releases that, then this small neonate has the mother's antibodies, and that's called acid immunity. Okay. All right. Questions about that?
What happens to that C receptor once it's in there? Would it pick up antibodies and just send them back to the extracellular fluid?
It's pulled off by constitutive endocytosis and actually sit back here, and then a sleight of hand that we don't understand goes back to the cell surface. Yeah, the typical membrane. Yeah. Okay. Good. Why is that not? Okay. Now, finally, we get to phagocytosis. This is, so you got huge cargoes, got large pieces of stuff that, like a bacterium that's being, this is actually not a bacterium, this is a red blood cell that's been mutated so that this macrophage, or whatever, I think it is a macrophage, sees it as foreign. And that little thing that's creeping up, oh, so this is the red blood cell, and the thing that's creeping up around that, that's the plasma membrane. But it's not going in, it's going out, right? These are arms. So these are pseudopods. So that's the big difference is you have pseudopods, and this is what's going on here. Pseudopods that go out instead of things coming in. So these are large extensions, and what tells the plasma membrane to do that is because the phosphorylation states of our old friend, the PI kinase system, phosphorylates this and turns it into phosphatidylinositol 3,4,5 and creates this, it tells the actin cytoskeleton to polymerize. That's what's causing the arms is the actin cytoskeleton. And what tells it to do that is the change in the cytoplasmic phosphatidylinositol phosphorylation state. And we know this rule. If you mess with the lipids, you're going to tell it to do various things. Why? Because a different set of proteins binds to this and recruits actin that binds to this. So you get these arms projecting out. These are called pseudopods, and eventually this is going to fuse with that. So it's like two vesicles fusing up here at the top. This is a single membrane-bound pseudophagosome, right, when it comes in. The cargo, this membrane of the cargo doesn't count. That doesn't belong to us. So there's one eukaryotic membrane around. So it's not like the autophagosome. Keep that separate. Okay. You're going to bring it in. Where does it go? So you bring it in. It's called an autophagosome. Where does it go next? Does it follow our rule for platinum coating? No, because it's not platinum coated. So it doesn't go to the early endosome. Besides, it's too huge. It goes to the late endosome, fuses with it, and then it becomes North Korea. Okay? And it gets destroyed. Okay, so, and this is a picture showing you, in case you didn't believe me, it does go into the cell. Okay, so now we've done all these things. The last one we have is one pathway. It's one of those places you can go when you get to the Trans-Golgi network. Remember, the Trans-Golgi network is a way station, sending things different places. Now we want to send stuff into what's called the regulated secretory pathway. And that's where all the really exciting things like hormones, growth hormone, insulin, it's where the GLUT4 storage vesicles that sit there and wait for the insulin signal to tell them to deploy it. So that stuff is going to go that way. Right? So we have two things that are going. So we have stuff going directly. This is the default pathway. If you don't tell me anything else about where you're going, I'm going to assume that if you get here and there's no zip code, I'm going to put you in COP2 coded or some other mystery coding. COP1 is a possibility. That's the only one I'll require you to know for constitutive trafficking to the cell exterior. So no diversions. Do not pass go. Do not collect $200. You go straight to the plasma membrane and fuse and eject. So things that do that, it's like collagen and things that we completely always building the extracellular matrix. If you want to go and have it stop and wait for a signal, that's called the regulated secretory pathway. And you have a very specific name for those kinds of vesicles. They're not transport vesicles, they're secretory vesicles. Okay? So we want to go this way, and so there is some sort of signal that diverts it to the regulated secretory pathway. And that actually, the process begins in the Trans-Golgi network. There's a couple things going on there. One is that there's some proteins that cause selective aggregation of growth hormone and the cargoes that are going to go outside the cell. But remember, they're going to go here and stop, right? And so they actually undergo selective aggregation, and there's a couple of proteins called chromogranin A and B and secreted granin that actually collectively cause insulin to coalesce and form crystals, almost crystals. And then what happens is, let's see if there's any more. So this is what's going on here. And this is a little confusing because it looks like there is clathrin-mediated exocytosis going off the trans-Golgi. And that would make a lot of sense, except it doesn't happen. What actually happens is a process, let's see if we have a picture at all. Yeah, a process that resembles phagocytosis is driven by the actin cytoskeleton, and there's an arm that reaches up here and reaches up here and pinches it off. So the cargo, like insulin or growth hormone, has been selectively aggregated, and it excludes all the cargoes that are not going into the secretory pathway. So if you're a lysosomal enzyme or a collagen, or you're going this way, you're excluded from this aggregation. So selective aggregation means exactly that. So what you do is you pinch this off by a phagocytotic-like actin-driven mechanism, and you have a huge secretory vesicle. There's lots of cargo here. So this is very biosynthetic, conservative. You're getting lots of stuff packed in here. Okay? And you can actually see insulin crystals. So what happens is there's too many goodies here. So what happens is you retrieve all the goodies using clathrin-coated vesiculation. So that's why that clathrin is there, is to remove the excess materials like the aggregating proteins and put them back into the trans-Golgi for another round. And then that makes your secretory vesicle very compact, highly concentrated. So when this thing blows into the plasma membrane, you're going to dump out a packet of growth hormone or a packet of insulin. And signals are secreted in pulses, and there's a reason for that. I think my endocrinology, if you want to find out. So this is going to hit this, but this is going to sit here and wait for a message from outside. If this is insulin, what's the message it's waiting for? Insulin is released in response to bird songs. You eat, your blood sugar goes up. Actually, amino acids are also another thing. And so it's waiting. And so it actually has a way of deconvoluting the blood glucose actually into a closed potassium channel that causes calcium to burst into the cell. And then this thing, as you know, calcium, if it's is released, this thing will then fuse. You get the synaptic attachment out of the way. This thing fuses and you dump the insulin outside. Okay. So here's the signal. It could be, so if this is, for example, a skeletal muscle cell, what's packed in the secretory vesicle that you're waiting for? GLUT4, right? Because skeletal muscle is a major consumer of glucose after a meal. So what you're waiting for is not arrive, because the skeletal muscle cannot directly detect, it has no detection, it has no glucose sensor. The beta cell has the insulin, it has the glucose sensor, and it sends out the signal to bind to the insulin receptor. That's the next topic we're going to talk about, is signal transduction. Activates this by a lot of jiggery-pokery, and it tells this to release a pulse of calcium, and then it fuses and releases. The GLUT4 is not secreted. The GLUT4 goes where? It goes into the plasma membrane. Because anything that's in this membrane, if this undergoes exocytosis, is delivered into that membrane. Okay? And so this just shows that whole process. This thing pinches off. Then the clathrin coating is to retrieve goodies to make this very concentrated. And then this actually binds to the cytoskeleton and waits like a kid holding onto a balloon. And you phosphorylate that kid's arm to get it to be released so it can dock. And so we don't go into that detail in this class. Okay, we talked about docking and fusion. Okay, the other thing I said, there was two things that are going on in the trans-Golgi network. in the last couple of minutes here. Okay. The other thing is lots of these hormones or enzymes are in a pro state. So this is what insulin looks like. It's synthesized as this long snaky thing so that it forms the correct disulfide bridge connections. And once it gets into the trans-Golgi network, you need to chop off this part right here and that part. And this thing is called the C-peptide because it looks like a G. No, it's called the C-peptide, it looks like a C. And so this C lifts off and floats away. Now it's actually in the secretory vesicle, so this is the active insulin now. And that process it's just a protease called prohormone convertases that cleaves it here and here. That process begins in the trans-Golgi network and finishes in the mature secretory vesicle. And actually, we now know that insulin is secreted, but so is the C-peptide. And we think that that actually has its own effects. So it may be fairly soon that people will be taking insulin plus C-peptide. Okay, I think that is about it. Yeah, we got that. We are done.